This video is going to cover section 10.7, which is on innate and adaptive immunity. The lymphatic system works with the immune system to protect the body from pathogens, toxins, and other invaders. So immunity is a condition where the body is protected from various threats, be they pathogens, toxins, or cancer cells. And there are basically two types of immunity. First, we have innate immunity, and this is fully functional without any previous exposure to substances. So it just kicks in basically automatically uh, with any types of threats. The adaptive immunity, however, this is initiated and amplified only after specific recognition of substances. So we'll look at both of these. First, we'll have a look at innate immunity. And the mechanisms of innate immunity, they can be divided into basically four types, where we have physical and chemical barriers, we have an inflammatory response, we have phagocytes and natural killer cells, and we also have protective proteins. So we'll look at each one of these briefly. First, we have our physical barriers, where skin and mucous membranes lining the respiratory, digestive, and urinary tracts. They're physical barriers to pathogens, so they prevent pathogens from entering the body. Then we have chemical barriers, for example, oil glands on the skin. They secrete chemicals that weaken or kill certain bacteria. Also, the acidic pH of the stomach, it kills many types of bacteria or it inhibits their growth. Bacteria that reside in the intestine and other areas, they remove nutrients and block binding sites that could be used by pathogens as well. So that's when we talk about the natural bacteria that exists within the digestive tract and other areas. So their presence actually blocks the binding sites that could be used by other pathogens, and so that's why the normal bacteria that exists is it's definitely required, and it's there for our health actually. So the inflammatory response then, inflammatory response, it's a sequence of events that's initiated when there's some kind of a damage to tissue, be that physical trauma or chemical agents or pathogens. So inflamed areas, they have generally four signs that they will exhibit. For example, redness, heat, swelling, and pain. This is mostly due to the capillary changes in the damaged areas, which we will talk about in just a minute. There's at least three types of cells in the skin and connective tissue that play a role in the inflammatory response. We have the mast cells, we have the macrophages, and we have the dendritic cells. So here's an illustration as to how that all occurs. So when you have some kind of an injury to the tissue, then the injured tissue cells and the mast cells releases histamine, which causes the capillaries then to dilate and increases the blood flow to the area. Then you have resident macrophages and dendritic cells, which phagocytize the pathogens, and they release cytokines then, which stimulates the inflammatory reaction. Also, neutrophils and monocytes, which become macrophages, they can squeeze through the capillary wall, and they can then attack the pathogens. Then, if blood vessels are damaged, then blood clotting, it will wall off the capillary and prevent blood loss. So this is quite a good summary of the inflammatory response. So the mast cells, they reside in the skin, the lungs, and the intestinal tract, and they respond to damage by releasing histamine. And histamine causes the capillaries in that area to dilate and become more permeable as well, which allows fluids to escape to the tissues, and that causes the swelling. The swollen area, it stimulates the free nerve endings, and that actually causes the pain. So you increase the blood flow to that area, and that causes the skin to redden and to feel warm as well. The macrophages and the dendritic cells, these both are phagocytic cells, and they release various pro-inflammatory cytokines, and these are chemical messengers that influence other immune cells. For example, interleukin 8, this attracts other immune cells to the scene, and a colony stimulating factor, and this actually causes bone marrow to produce more white blood cells. The phagocytes, they engulf the pathogens into an endocytic vesicle, which fuses with a lysosome in the cell, and then the pathogen is destroyed by the compounds in the lysosome. It includes neutrophils and monocytes. So the neutrophils are generally the first white blood cells to enter an inflamed area. And these are often the ones that accumulate in very large numbers and to form the pus, which often occurs with infection or damaged tissue. The monocytes, they migrate from blood and they differentiate into macrophages in tissues if an inflammatory reaction continues. We have natural killer cells. These are large granular cells that kill some virus-infected and cancer cells by actual cell-to-cell contact. And these cells, they induce target cells to undergo apoptosis, which is a fancy name for cell death. So what they do is they seek out and they kill cells that lack a type of a self marker or self molecule called MHC1 on their surface. And some virus-infected and cancer cells, they lack MHC1, which makes them susceptible to destruction by the natural killer cells. So that brings us to adaptive immunity, and adaptive immunity is activated when innate defenses, they fail to prevent an infection. So the adaptive immune system, it recognizes, responds to, and it usually eliminates antigens from the body. Now, what do we mean by an antigen? An antigen is any molecule that stimulates an adaptive immune response. So that would be, for example, like a protein on an invading cell or a cancer cell. So they would have antigens on them that stimulates an adaptive immune response. It takes actually a few days. So this is a process that doesn't kick in immediately. It does take a few days in order for it to become fully functional, but it can actually last for many years, even decades. So the adaptive immune system, it depends on the activity of B cells and T cells. Both of these cells, they recognize antigens because they have specific antigen receptors on them. And each lymphocyte actually only has one type of receptor. However, we have a large diversity of antigen receptors on B and T cells. We actually have millions of different kinds of these. And so we have B cells and T cells to cover just about any possible antigen that we may encounter. So the defense by the B cells, and this would also be thought of as antibody-mediated immunity. The B cells, they're activated in lymph node or the spleen, where the receptors bind to specific antigens. The cytokines, which are secreted by the T cells, they stimulate B cells then to divide. And most of the B cells, when they divide, they become plasma cells. And these secrete antibodies, and the antibodies are the secreted form of the receptor of the B cell that was activated. Some cells become memory B cells, which allows for long-term immunity. So that means then if the same antigen were to ever again enter then the memory B cells would divide and give rise to more plasma cells that can produce this antibody against the antigen. So it would be a much quicker response if that same antigen were to be presented in the future. So the antibody structure then, antibodies are also known as immunoglobulins, and they have kind of a Y shape to them, and they consist of two arms made of polypeptides. And each has a heavy peptide chain and a long peptide chain, and each of these chains then has a constant region and a variable region. So the variable region would differ from antibody to antibody, and this would be the amino acid sequence between the antibodies to form the antigen binding site. So each one differs in that regard for a specific antigen. So here's a diagram of that, and you can see here that we have the heavy chain and the light chain, and each of those, the heavy chain and the light chain, has a variable region which accounts for the different amino acid sequence, which will allow it then to bind with a specific antigen. So antigens then, they combine with an antibody at the antigen binding site in a lock and key type of a manner. So these are kind of shape dependent. So the antigen antibody reactions, they can result in immune complex, and the immune complexes then, they mark the antigens for destruction by neutrophil or macrophage. And the antibodies can also neutralize toxins by preventing them from binding to cells. So the antibody then binds with the toxins, and then their binding sites doesn't allow them to bind to cells then. So the T cells and cell-mediated immunity. So the defense by T cells. T cells have a unique T cell receptor, but they cannot recognize antigens on their own. So they require antigens to be presented to the receptors by an MHC protein on the surface of another cell. And there are two types of T cells. We have helper T cells and we have cytotoxic T cells. The helper T cells, they only recognize antigens that are presented by specialized antigen presenting cells with MHC class II proteins on their surface. The cytotoxic T cells, they only recognize antigens presented by various cells with MHC class I proteins on their surface. So some T cells actually become memory T cells, and again, they will live for many years and can quickly initiate an immune response to an antigen that's previously been present in the body. So then it'll therefore initiate a much faster response to that particular antigen if it ever gets encountered again. So with the cell-mediated immunity, the activated T cells bound to a virus infected or cancer cell, it releases a perforin, and that forms pores in the plasma membranes in the abnormal cell, allowing enzymes called granzymes to enter the target cell and induce cell death. And here is an electron micrograph of a cell that's just been targeted by a cytotoxic T cell, and it's undergoing cell death. So there's a lot of details in this, and there's actually a couple of great videos in the course resource as well. So you'll probably want to watch those a number of times to make sure that you get all of the details of these processes understood.